Hello everyone, I'm Lindsay Overmyer. I'm the Ramsey County Ag Agent with NDSU Extension. Today I'm presenting on CSI, and that is Crop Scene Investigation. So what is CSA? Crop Scene Investigation. So it's really a systematic way to go about diagnosing problems in the field. This was developed from multiple professors at the University of Nebraska-Lincoln, and currently there is a practical manual of field crop diagnostics book. It's in preparation right now, and hopefully further in later on here in 2022, it will be coming out. So there are 7 steps in this diagnostic process. We have the first step where you're gathering field history. The second is field scale observation. Then we look at the plant scale observation, the leaf scale observation, and what are some additional questions that we need to be answered is step 5. Finally, we come up with a diagnosis. And then do we need other additional tests to confirm this diagnosis is that 7th step. So the first step is gathering your field history. So this can be multiple components. So the first component we have are cultural practices, pest management, soil management, water management, and looking at these a little bit more in depth. Under cultural practices, we have what is the operator history? What is the field layout? This crop rotation history. We have planting equipment, which can vary between farmers. What are the variety that was planted this year? What are the characteristics, the strong points of that variety? And also then looking at the yield history. Next, moving on into the pest management, what some of these pests that can be in the field where could be pathogens or diseases, arthropods, weeds, wildlife, and have there been any pesticide applications on any of those on this field? And what equipment are they using? Are there different practices? Is it a weekly preventive spray, or it just depends upon the crop in the field. The next one we have here are soil management. This can be really, thinking about the field. What is the slope of the field? Is there topography? Is there a drainage way in the middle of the field, or so forth? Are there sloughs like here in North Dakota? What is the soil type? Different physical characteristics of the soil, crop fertility, and also what are the tillage practices, or what's the history behind it? Or is this conventional till? Is this no-till? Is this reduced till? And so forth. And finally here, the last one is water management. Again, kind of going back to thinking, is there a slope in the field which makes water run a certain way? Is there a drainage way in the middle of the field? And, it's precipitation. Has it had a lot in the past year, or are we in a drought? There's different irrigation types. If it's a center pivot, linear, and all of that, all of the water is deeply connected to the soil type. So on the right, you have been seeing multiple images of a field scenario. We kind of diving into this field. So we arrive, this is a scenario that occurred in August of 2019. So we're arriving at the field, just kind of doing some late field season checkups. What are we seeing? And this is some of the field history that we had collected and that we saw. So, and understanding some of that field history. So some of these cultural practices, we know it's a soybean wheat rotation. It is in a reduced to no-till system. We have dicamba resistant soybean variety, which is planted, as you can see in the pictures here on the back. There's a little topography change in the field, but there is some. And we also know we don't actually know much of the pest management about this season. We do know that we have water management. It is a rain-fed field, and we do see a drainage ditch in the field. So we'll kind of be diving into these pictures, and we roll up to the field. Now what do we see, and how do we go about it? So this is just step one of understanding what this field history is. Now, looking into more and making those observations, this is your field scale observation. And what are we seeing? So, understand field scale observation, we need to understand a distribution pattern. And there are three main distribution patterns. And this is all relative to, we could potentially see a pattern, but what is the scale at which we are seeing? So to first look at one distribution pattern is the uniform distribution. And this is usually indicating a man-made or abiotic problem. This could be equipment-associated damage or seed-borne disease, for example. And to kind of further explain this picture here, we have, so the squares indicating a field, all the green rows or circles are referring to crops, and the stars are then trying to show the distribution pattern. So all the stars are evenly distributed in this image. Next distribution we have is the random distribution. So you can see that the stars, in this mock field, there's no rhyme or reason to any of them, that they're just over somewhere. We have more here on the south side than on the north side. And these are really thinking about scattered symptoms. And an example of this could be insect infestation or a foliar disease that there is no rhyme or reason. It's just there. And really on this random distribution, the scale will determine the pattern, because if you're only looking at the south half of this field, maybe, it could be, to me it's still random, but if you maybe only look at, a certain part of the field, you could think that it would be a uniform distribution. So really thinking about that scale of the entirety of the field, or are there neighboring fields that are involved in this problem? The last one we have is an aggregated distribution. This also could be known as a clump distribution. You can see that these stars are very much grouped together, clumped, and have different, and that's their distribution. So the damage or the symptoms or injury are occurring in different pockets where the stars are grouped together. This is usually associated with an element in the field, such as the topography, soil types, and examples of these can be an insect infestation, a soil-borne disease, or potentially even a herbicide carryover that's occurring in low spots where residue is building up and staying put. Now, this is our field that occurred here in around Jamestown area, and this is a late August, and these are the field scale observation. And these pictures are from a drone, so to show this and try to orientate you to this field, this side of the field is the north part, so this is why the compass is showing north, and we can see here that there is no soybeans, a thin stand, and a little bit better of a stand per se, and into our stand that looks pretty good. And our next picture, so these fields are right beside each other, so this lane is the lane in this photo as well. So now looking at the adjacent field, we can see that there's like this big old drainage ditch in here. Now there's some other spots. This could also be kind of thinking maybe a saline area, but this is definitely more of a drainage way where there's, less the stand is reduced. There's less green along here where you get into the middle of this drainage way, and there's no green, so there's no stand. So again, these are fields side by side to kind of orientate yourself a little bit better. So that first picture here is in this northeast corner of this field. We have this field lane that is in between these fields, and that is where this next image is from. Next up on the systematic diagnosis is step 3, and this is looking at that plant scale observation. So for this plant scale, we have it closer up pictures, and so again, the left picture is more on that north east side of the one field. So each, on the image on the left is corresponding to that oh, we can't go back to this. So it's corresponding to the northeast section of that field, and then this is, near the picture on the right is the drainage way area looking. So again, like I pointed out, we have that, there's no soybeans here, thin stand, and then we get into our better looking soybeans here. And then here, the image on the right, you can see that we have soybeans that are good stand, and then we get into this area where we see that there's less stand, and they kind of looked maybe, they don't look this normal green like the leaves are, out, spread out, like fully expanded, versus these look, something looks off here. So again, orienting yourself to these pictures, we're going to really focus on this area because we're driving down this lane and we're looking, we're like, oh, something looks different here. So this is looking directly east into the drainage area. So this is the drainage way right here. And we can see that there's kind of this weird, line. We see good soybeans here. These soybeans look different. Something's off. They look sick. Again, we kind of see a different line here where, some of these don't look so good, and we have soybeans here looking pretty good. So that's looking east directly into that drainage ditch area. Now, if we're turning and looking north, we can see that, again, a clear difference in this line. Really, we have healthy soybeans on the right, poor soybeans looking on the left. This is the edge of the field, and that field lane is just right here. So also with that plant scale observation that we're wanting to look at, is the damage, is that injury, the symptom, is this on the above or below ground of the plant, or is this also on the new or old growth? So we can tell that all of this is on the is on the new growth, because that's the top of the plant, where all of the new, where the new leaves are forming into those different trifoliates. Unfortunately, in this scenario, we don't have any below ground images of the roots, so sorry about that. So just, again, looking at a little bit closer look, healthy. These soybeans also look stunted. The inner nodes are closer together, and they're shorter. So both looking from the north and south of that drainage area. And again, on this side, we see that the field edge is right here, and the field lane is just right beside this on this image. All right, so kind of a summary of the plant scale observation. We have dead plants in the drainage way. There's stunted plants along the drainage way or in lower areas of the field. Plants that are further away from the drainage area are appear to be normal, they're healthy, and unfortunately there is no observation of the roots. So these are our sorting plants here on the right. Our next step in this observation is our leaf scale. So where is the distribution occurring on the plant? And now I'm going to specifically be looking at the leaves. We can see that these leaves are, they almost look like they have blisters. They're malformed or deformed. They, kind of this drawstring effect here on the leaf tip. We also can see a variety of symptoms in this field. We see that image here on the right. They're even more cupped, puckered up versus the one on the right. And again, you can tell that these look like they're a little bit bigger versus these must be closer to that drainage area because they're even shorter. So the summary, we have some leaves that are cupped, crinkled, puckered, strap-shaped, malformed. On all of those leaves are in the upper canopy. So it's looking at more of that new growth. And there's also some insect defoliation out there. We also seen some of these thistle caterpillars and some of this defoliation occurring in the leaves. And this is again in the upper canopy. But the main one that we kind of want to be thinking about is that what is causing these leaves to be so maldeformed. So now after we're gathering all of our observations, are there additional questions that we need to look at? So kind of what is the next step? One of the first questions I may ask is, what is the soil type of the field? That can kind of relate to this aggregated distribution. So we have that first issue in the field that's happening in this area. So we knew that this soil type is G143A. It's a Barnes CV loam. We have 3 to 6% slope here in that area. Now the other part of the field that we're having an issue is that GTA, and this is a Tonka silt loam where it's a 0 to 1% slope. So maybe these soil types can be involved in this issue of what we're seeing. And an very important question is what was last year's herbicide program? We know that these soybeans are dicamba resistant, but what was last year's? Could that have had any effect in this situation? So it was a field match. So going back to, it's a soybean wheat rotation. We have a wide match, which was used last year on the wheat, and it is clopyralid. It's a growth regulator. Site of action is 4. And then kind of further thinking about it is like, what was the growth stage of when when white match was applied to the wheat? And we're looking at a vegetative stage, so probably May to June in this sense, in this field. Another important question is to understand about, how herbicides react in the soil, how herbicides may kind of hang in the soil versus being degraded, is thinking about how much rainfall there has been since the application. And also, what was the timing of the rainfall application? Or, sorry, what was the timing of the rainfall? Both of these information can be looked at and found in NDON, and that will be a powerful tool in trying to help solve diagnostic issues. So now we have gathered our field history, we have our observations, we're thinking about those other questions that need to be asked, and so now we're coming down to that final stage of diagnosing what has happened at this crop scene. We have defoliation in the upper canopy. Clearly, we saw a thistle caterpillar in that image. So we know that there's insects feeding in the upper canopy. Is this a really big deal? No, because this is not exceeding the threshold. But we're gonna, so it's not exceeding the threshold. We know that soybean defoliation threshold is lower when we're getting into that reproductive growth stage. So that's 20% defoliation versus our 30% defoliation when they're younger, because they're still going to be making new leaves. The other scenario, like we said, we know that it was clopyralid, which was sprayed from last year's herbicide program. And this is actually clopyralid, and some of this residue is concentrating into the lower areas or in the drainage area of the field due to the water movement. We know that we saw that for sure in that all the images on the right had that drainage ditch along there, and that is definitely, which is going to have more water movement, residue that's going to come along with the water. So that is why the soybeans near those locations of that drainage area are displaying more of this plant growth regulator symptomology. And really, to understand a little bit more about this plant growth regulators, again, their site of action number 4. They are synthetic auxin or auxin inhibitor compounds. So they are they're mobile in the plant. So they're moving through the xylem and the phloem. They're moving to the actively growing areas, so where cells are dividing. And this is where since it is a synthetic auxin or an auxin inhibitor, this is disrupting or imbalancing of the hormones, which is, messing up, disrupting that cell division and that protein synthesis within the plant. So this is why it is occurring in the new growth. And because these symptoms are occurring in the new growth because there's a higher concentration of protein synthesis cell division occurring in these upper leaves of the soybeans. So this is why we're seeing this symptomology in the plant. And this is the diagnosis that we're having. And are there any additional tests to confirm this diagnosis? So in this regard, both of these fields are owned by the same farmer. We can know that the diagnosis is because of last year's clopyralid that was sprayed on the wheat, and this is just herbicide carryover. So in this regard, we're not going to do anything else, but if this was, these two fields were different farmers, or maybe, other things where we would want to for sure confirm our diagnosis, this would be our 7th step. And to confirm, we saw insects in the field. They're present on the leaves that had defoliation. Again, we do not need to treat them because they were not exceeding the threshold to treat. And the important part, because there's a lot more damage with the growth regulator symptoms, would be screening of active ingredients with the plant growth regulators. I did look this up. You can do a panel of plant growth regulators, and that is going to cost you $212 at South Dakota Ag Lab. And what you would need for that sample is a gallon-sized bag of effective vegetation or 4 cups of soil for them to have enough material to perform the assay. So in summary of our crop scene investigation, we were gathering that field history. It's a wheat, soybean, wheat rotation. We were seeing our field scale observation. It is a clumped or aggregated distribution, as we saw in both of these fields here and there. Our plant scale, we had, our upper canopy, our new growth that was more affected, and we had stunted plants. And then we got down to our leaf scale where we had these puckered, malformed leaves. finally, we had those additional questions of really, what was last year's herbicide program? Could this affected this year's? We made that diagnosis of our plant growth regulator clopyralid as a herbicide carryover, and we then have the option to confirm additional tests. So that is the 7-step way to diagnose things, and this is just a way to investigate a field scenario systematically, that we're looking at all things and having an open mind of what could this be. And I just want to thank you for listening to me. So if you have any questions, you can email me at lindsay.overmyer@ndsu.edu. So I thank you for your time and have a great rest of the day.